I'm in section 8.4, and if you're listening to 8.4 before 8.5, I would recommend that you stop here and go back and listen to 8.5 first. I have them out of order on purpose. So listen to video 8.5 before you do 8.4. So here we are on 8.4, and it says compare vertical gene transfer and horizontal gene transfer, and explain why horizontal gene transfer is important in bacteria. So what I mean, so remember, our genes are the instructions on how to make each protein, right? And so we have to transfer our genetic material, our genes, our genome, whatever, from one generation to the next. So here's a bacterial cell, there's the chromosome, and here's that DNA replication, chromosome being copied into two. The cell divides into two, and now we have two daughter cells. And so this is what happens when cells divide. We call this vertical transfer of genes. The genes in one generation are copied, and they're in the daughter cells. So the parent cells and the daughter cells have the same genetic material or shared genetic material. So we all have, we've all vertically received genes from our parents. So the genetic material from one generation is passed down to the next. We call that vertical transfer of genes. All organisms have to do that. But there's something else called horizontal transfer of genes. And what that means is transferring DNA between individuals of the same generation. And so the way I like to think about it is I have very straight hair. And I would love to get one of those genes that helps me have curly hair, because that's a genetic trait. So if I inherited a gene for curly hair, I might be able to have curly hair. But the only way I could get that, since I didn't get it from my parents, I'd have to get it from someone else. So I'd love to walk up to somebody else and say, can you give me some genes for curly hair? And they transfer me some of their DNA, and now all of a sudden I have curly hair. We would call that horizontal gene transfer. Now, humans don't do that, except we are trying to do that in the laboratory. And we're trying to do that in the laboratory to to cure diseases like cystic fibrosis or sickle cell anemia, where we can replace damaged genes or genes that aren't functioning normally with healthy genes. So this is something that you hear about with genetic engineering, with manipulating genes in humans. And it will continue to be something you will hear of in your lifetime. It's a big deal. But we don't do that naturally. We can't horizontally transfer genes naturally in humans. But bacteria, a lot of bacteria can do it. So what these are, and this picture is also in your study guide, is they're just pictures of some of the ways that genes can be transferred horizontally in bacteria. And in this example, what they're showing you is if a virus infects, you don't have to know the difference between these either. I just want to give you some examples. But here's a bacterial virus, a bacteriophage. If the bacteriophage infects the host, it can pick up a little bit of host DNA. So what they're showing you is here's a bacterial cell, a bacteriophage, a virus, that picked up some DNA from its host cell. And when it goes to infect another bacterial cell, it can actually release some of the DNA from the previous cell into here. So you can add some new DNA into this bacterial cell. This bacterial cell actually received some DNA from that bacterial cell. So that would be horizontal transfer. Here's another one. Some bacteria can actually what they call conjugate. This is the closest you'll see to bacterial sex. Not all bacteria do this, but some can. And here's where that plasmid comes in. Sometimes during conjugation, DNA from one bacterial cell can be transferred to another. So here we are transferring DNA between two cells in the same generation. They're not parent to daughter, but they're two cells in the same generation. Here's another thing that's called transformation, that when bacteria die, they just, they leave their parts laying around. And so there might be a little bit of DNA in the soil where the bacteria were when they died. And so some bacteria can actually pick up that DNA and incorporate it into their cells. Now, why would a bacterial cell want to do this? Why would they want to pick up DNA horizontally? Well, the same reason why I would want to get a curly hair gene, to get some new traits that allow them to have certain characteristics. And a big one where this becomes important is in antibiotic resistance. So let's say this bacterium is resistant to an antibiotic and it has the genetic material that makes it antibiotic resistant. A virus can actually transfer those antibiotic resistance genes to other bacteria. Here in this case, this bacteria is resistant to an antibiotic. On that plasmid, it's got an antibiotic resistant gene. It can transfer that antibiotic resistance to a cell that wasn't able to do that before. So this is a big way that that more and more bacteria become antibiotic resistant. That they don't just inherit antibiotic resistance genes from their parent cells, they inherit from bacteria from the same generation. So this is a big way that antibiotic resistance is propagated. I think I'm going to stop there because I think the last part can take a little bit longer.